So everybody ready to talk about the coolest viruses we're going to talk about all term? Yes, definitely. Well, hottest virus. Sorry, the hottest viruses we'll talk about all term. So, but first, really quickly, everyone filled out their online evaluations? Yes? No? You're going to? Yes? You're all coming to graduation? Yes? Even if I haven't posted the grades by then? That's it, yeah. Unfortunately, we don't offer A+ here at PSU. So, otherwise, of course, everyone would have one. Assuming they answer the next two clicker questions correctly. We ready to do that? Almost the last clicker questions of the term. So Dr. Oredas will not have clicker questions on Wednesday, but we will have those on Friday before our review. Okay? So no clickers. He's not going to be recording his lecture, so talk to people nicely about trying to record those as well. So, and as long as I get recordings, I'm happy to post them appropriately. They won't be on YouTube. Okay, other questions, comments, worries at this point? Other than, let's see, we're going to talk about these viruses. We're going to talk about these viruses, some even cooler ones that are not on here. But first we need to talk about the giant viruses and ask clicker questions about them. So the white, I noticed this when I put it together, white cliffs of Dover, not just one, are probably due to a virus most similar to Mimivirus, Ebola, E. hux virus, PBCV1, or measles. I can see how people vote actually up here, so I actually do get feedback. It's quite amusing. Ten. 5, 4, 3, 2, 1. Do we have a consensus? Yes, we have a consensus. So nobody liked Ebola, nobody liked measles. Mimivirus and PBCV1 are definitely giant viruses that are quite closely related to the viruses that are infecting these coccolithophores, but they're nowhere near as closely related to the E. hux virus. So yes, that is correct. Hallelujah. So C it is. And our last question for today. The virus with the largest virion known to date is, we're not quite sure where those quotes came from, Mimivirus, Ebola, Megavirus, Pandoravirus, or Pithovirus. 5. Okay, so which one has the largest genome? Pandora virus. Which one has the largest virion? Pithovirus. Ebola is really long and skinny, but that's about the only thing. Actually, the Ebola virion is longer than either mimivirus or megavirus in terms of the absolute diameter of what it is, but still way smaller than any of these absolutely insane virions that we have here at the end. So yes, pithovirus is the largest so far. I haven't noticed anything in the literature that's come up since then, but there may well be some that I haven't noticed yet, or someone will be announcing at the meeting that I'm going to be talking at in beautiful London, Ontario, in July. So the American Society for Virology meeting. Just got the schedule for that. Yes, London, Ontario, or Texas, Paris, Texas, etc. So archaeal viruses. One day, maybe before I retire, I'll actually teach a whole course on these things. But you have to do this one first and then all the other courses as well. So these are almost all double-stranded DNA viruses. Double strands, of course, in quotes, because a number of these are, in fact, very similar to the genomes that we looked at for the pox viruses and PBCV1 with these covalently closed ends. So what do you really mean by double-stranded DNA virus? A lot of the viruses of the so-called Euryarchaea, there are two main branches of Archaea, the broad eukaryotes, which actually have viruses that look mostly like bacterial viruses or bacteriophage, and then those that are nearest and dearest to me and everybody in my lab are these from the Archaea, which have a very unique virion morphology and also completely unique genomes. And these are 5 of the cartoons from the Viral Zone website. Also a really good place to go and look in terms of review of the different kinds of viruses, in terms of something that's coming up next week from tomorrow at 8 a.m., by the way, not 9, 8. All of these Archaeal viruses so far have been found at very high temperature environments. They're also the hot viruses. So what I'm going to talk about, again, mostly these interesting viruses of Archaea. We'll talk a little bit about the viruses of the Euryarchaea, just because they're there and all of Archaea. So just a quick background. So there's Archaea, bacteria, and eukarya. So this is like one lecture talking about all of the viruses of eukaryotes, right? So clearly we're going to be compacting stuff a lot here. Of the viruses of Archaea, there are 1, 2, 3, 4, 5, 6, 7. Actually, I think there are about 9, maybe as many as 10 virus families. Each of the chapters in the book is usually one virus family. So again, gives you an idea of the diversity which are there. Spend most of the time talking about the Fuselloviruses, which are these guys up here. Fusello in Latin means spindle. I think you should really call these the citronoviruses because they look much more like lemons. Then the Rudiviruses and Lipotrix viruses. These are long rod-shaped viruses. The Globuloviruses look like globes. Ampuloviruses really do look like bottles. B. cauda is 2 tails. And the Turriviridae are these guys down here at the bottom with these amazing turrets at the 5-fold axes of symmetry. Why do I have a chip on my shoulder about archaeal viruses? This is the Virosphere, published by the ICTV. You remember how much I like the ICTV. 2005, all of these viruses here on the outside are viruses of eukarya and bacteria. And then we've got this one little slice right over here for viruses of Archaea. Again, not doing justice whatsoever to what's going on there. I think this is a much better way of thinking about viruses of Archaea, because Archaea are one of the 3 major groups of cellular life. We've got eukarya, represented by all animals right here, Homo being us, all plants right here, and all fungi right here. There's actually a lot of diversity in eukarya that a lot of people don't think about. Things like Giardia and Trichomonas, lots of single-celled eukaryotes. Very little has been studied in terms of the viruses there, but almost all of these are infected by viruses. All bacteria, E. coli, etc., all infected by viruses. So it's not surprising that most of the archaea, all represented here by little bars, because this is the small subunit RNA sequence relationship between all of these organisms, all very different relative to each other. Each of these groups have viruses that are associated with them. One of the reasons that we're so interested in studying the viruses of archaea is they have a distinct similarity in terms of how they function, particularly at the levels of transcription, translation, replication, which of course are all things that we've talked about in terms of viruses so far this term. Turns out that all those machineries are very similar to eukaryotes. So they're a nice way of understanding some very basics about sort of the evolution of some of these machineries, and may even allow us some insight into how machineries work in this one particular group of organisms that we're very interested in. They do thrive in extreme environments. This is what got me excited about these viruses and the organisms they infect, because I can't live in boiling acid, but these guys can. So really quite impressive. They do have very different lipids. These are ether-linked lipids. Some of them, in fact, make monolayers across membranes rather than bilayers, which is a really fascinating adaptation. And they may be similar to ancestral organisms. People talk about the last universal cellular ancestor being here, sort of the base of one of these evolutionary trees, and the archaea are really close to that. There are all kinds of caveats about how similar those are at all. We're going to mostly talk about viruses infecting Sulfolobus because it's really easy to grow. Ask people here who are in my lab. And yeah, you are here, relatively speaking. Why are these so bizarre? Well, they are. We don't know why, but they are. So This Week in Virology always says you should never ask the why question in biology. You should always ask the what question. So what's there? The virions of these archaeal viruses, with a few exceptions, this one actually looks a lot like some of the bacteriophage. These guys all look extremely different. Probably the most different of those are these viruses, the Acidianus bottle-shaped viruses. These are the Ampullaviruses. They really do kind of look like a champagne bottle at one end with these birthday candles at the other end. Really amazing virion structure, completely unprecedented. Another one of my not quite completely favorites are these Acidianus filamentous viruses. We'll talk about these in more detail a bit later on. This scale bar is 100 nanometers. So that means that this virion is almost 2 microns in length, which is getting close in terms of size to some of the things like the pithovirus, etc. Only these are really, really skinny, and they've got these amazing claw-like structures at the end. This is the receptor interaction as far as we can tell. Again, the bar is 100 nanometers here, really tiny claw-like structures which are there. You also have some sort of icosahedral-like viruses, some clearly icosahedral-like viruses, but T equals 31, very bizarre kind of icosahedral symmetry there. And then our favorite here, the SSV viruses as well. So we'll start talking about the boring viruses. These are the viruses of the Euryarchaea. Why are they boring? Well, actually, they're not boring at all, but we don't work on them. These look a lot like a lot of bacteriophage. So this particular one here infects methanogens, which are organisms that are producing methane. Again, maybe related to some of the very first cellular organisms on the planet. These guys have virions that look a lot like things like lambda. You also have a few viruses that are infecting some of the methanogens. Actually, let's see, it's the one that's not on here. Have some spindles as well, lemon-shaped particles that are infecting them. Another big group of archaea are the extreme halophiles. Anybody who's flown into San Francisco, when you come in, if you look out the window, you'll see all these purple spots are actually really big areas of where the salt pans are. They're drying out salt to make sea salt. All the purple there comes from these extremely halophilic archaea. So one year when I was teaching microbiology, we took a field trip and got on a small plane and flew around. It was like the coolest field trip ever. So the extreme halophiles, these guys have a number of viruses that infect them that also look a lot like bacteriophage. But more recently, partly in fact due to some of the work that we were doing looking at these lemon-shaped particles, people found some lemon-shaped particles that infect these organisms, as well as some viruses which turned out to actually look extremely like the STIV, these turreted viruses as well. There are some extreme thermophiles that are in this group of organisms called the Euryarchaea. Dr. Bartlett's lab works on Pyrococcus. These are organisms that grow at 100 degrees Celsius plus, and they also have some viruses that are associated with them, which are shown down here at the bottom. And we have a visiting scientist in our lab right now from France who works closely together with people who have been working on these viruses. Probably the best understood of those viruses that infect the euryarchaeotes is Phi-H1. That's this guy on the left. This guy on the right is the virion from a bacteriophage. I challenge you to see much difference even at this really crummy resolution here that I copied these slides from. Look extremely similar to bacteriophage P2, which, again, we haven't talked about this particular bacteriophage before, but it's got a rigid, stalked tail and a nice icosahedral symmetric head on it. Curiously enough, the genome of this virus looks quite different than this one. It looks a lot like lambda. It's got a linear genome that circularizes once it comes inside the cell. Divergent transcriptional regulators, probably regulated by anti-termination, and maybe also some UV induction, but how that is working is nowhere near as well understood as what's going on with lambda. So that's basically it for these boring viruses. The exciting ones you find in places like this. So this is me collecting samples with a long pole. This is a volcanic hot spring in Yellowstone National Park, 85 degrees Celsius, pH of 3.5. It really does look like tomato soup. Wouldn't recommend drinking it, but this is exactly the right kind of environment for the viruses of Sulfolobus and other Archaea in general. Some of you may get a good idea how well faculty members are paid. This picture was taken in 2000, and you may recognize this fleece. So what you find in these environments most commonly are these lemon-shaped viruses. And so for the next 10, 15 minutes, I'm mostly going to talk about these lemon-shaped viruses, or the Fuselloviruses. Here's one virion down here at the bottom. Really does have this sort of lemon shape with a short tail at one end. You very often find these associated with cellular debris. So this is probably a bit of blebbed off cell here. All of these virus particles are associated with it. I'm actually quite proud of this picture. It's one of the first ever TEM micrographs that I ever took. This bar here is 100 nanometers, and that shows you that at least in negative stain, these are about 60 nanometers across by about 90 nanometers long. Inside this virion is a double-stranded circular DNA, but the really amazing thing about it is it's positively supercoiled. And this was, in fact, the first ever example of stably positively supercoiled DNA ever to be identified. Now, why positive supercoiling? That's overwinding the DNA. Why would you want to overwind the DNA? To compact it and to keep it stable at high temperatures, because these guys are in those bubbling hot springs like the one I just showed you on the last slide. So positive supercoiling makes sense in the case of these viruses, and it turns out plasmids in these organisms also have positively supercoiled DNA genomes. And we'll see a little bit later on some other examples of how that DNA is stabilized to these really extreme conditions. This genome integrates specifically into a site in the host genome, very similar to what happens with lambda bacteriophage. And it turns out there is, in fact, an integrase gene, which does this job, takes the genome and integrates it into the host genome. Also, like lambda, production of these virions is inducible by ultraviolet irradiation. And this particular virus was originally isolated from an organism found in Kyushu, a place called Beppu Onsen in southern Japan, an organism called Sulfolobus shibata. So what does that genome look like? Here's one of our favorite maps here. Each of the arrows around the outside represents one of the genes. I already told you about the viral integrase. This is a protein with sequence similarity to a whole family of integrase genes that you find throughout lots of different viruses. One interesting difference between this integrase and something like the lambda integrase is the attachment site, where that integrase gene product binds to integrate the genome is in the integrase gene. And so what that means is when you integrate the genome, you disrupt the integrase gene. So this integration seems to be a one-way ticket. You integrate into the genome, that's it. You're stuck in the genome. And so that was a really interesting discovery that was made quite early on, in fact, even before I started working on this particular virus. Another couple of things that were known before I started working on this were the gene products made by these three genes right here, isolated in red, highlighted in red, I should say. Again, incredibly creatively named VP1, VP3, and VP2. These are the proteins that you can identify if you purify a whole bunch of these guys and just look at them with STS-Page. You'll notice a bunch of other arrows that are present on here. We have about this much idea of what they're doing. So that's where this really stood when I started working on these viruses. I just noticed almost 20 years ago, which is kind of frightening. But I've discovered a few things since then. And a lot of that depends on a method which a PhD student of mine, Adam Clore, developed. And this has been picked up on by Eric Iverson, who's a PhD student who's almost done in my lab. What we did was we developed this thing we call long inverse PCR. And the way this works is we basically design two primers. Here are these two red arrows on the outside here. And then these primers, instead of facing toward each other, it's what you would usually think about in terms of PCR, is they face away from each other. But since this is a circular genome, they can amplify between these two. And then what you can do is hook these two ends together, and in that process, delete whatever gene you want to. And we started out deleting the integrase gene because that was the only one that actually looked like anything else in any of the databases, and we thought we knew what it was doing. So we deleted this gene, and to our surprise, the virus was perfectly happy. It didn't care about getting integrated. And so what that means is, and actually going back to where you have the integration site in the middle of that integrase gene, that probably means that that integration is not essential for virus function because you're messing up that integrase gene. So it turns out that these viruses do perfectly happily without having this particular integrase gene. And so it took Adam most of his PhD to figure out how to do this and make these deletions. Now we have this technique down. Eric has taken off with that and deleted a whole bunch more genes in the genome. And so everything here, which has a color, either green or red, is one of those genes that Eric has deleted using this same kind of technique. And the color coding here is basically you can knock it out and the virus is happy. It's green. You knock it out and the virus is unhappy. It's red. So this is, again, a large part of Eric's PhD thesis. Couple of things to notice on here is there's a bunch of green down here. And there's some more red up here, although strangely enough, a couple of green things as well. Even including some of those viral structural proteins, which we really didn't expect at all. So this is, again, most of Eric's thesis. but it took him anywhere between 2 to 3 to 6 months to knock out each of these genes. There are 34 of them here. I'm sure you've counted them already. He didn't want to take 34, let's see, 6 months per gene, 34 genes. 17 years to finish his PhD. Didn't like that. So we decided also to do a insertional mutagenesis. And this has been, again, through Eric, and then undergraduate student working in the lab, David Goodman, Valley Huang was a high school student, and then everybody who's in the recombinant DNA techniques lab has also been doing this. They've been analyzing each of these individual insertion mutants. Each of these arrows here represents one mutant, and then, just like the colors here of these arrows indicate whether this particular virus works with an insertion there as opposed to one over here. So again, similar kinds of patterns that we see. Now we've looked at most of the genes, and actually now we're up to all the genes in the genome. This is slightly out of date now. A whole bunch of green over here, a whole bunch of red over here, with these funky exceptions up the top here as well. So this has given us some good indication in the lab how these are working. The other way to try and figure out what each of these genes are doing is to let nature tell you what's going on with these. As I mentioned, you find these in volcanic hot springs. Turns out that there are volcanic hot springs all over the world, including Iceland, Yellowstone, and Kamchatka. And basically what we've been really fortunate to do, I didn't know that my job was to have to go to these places and collect samples. Talk about a cool job to have. I have to convince people to pay me to do it, which is a little bit harder. And then get the viruses from there and then just sequence their genomes and compare the genomes to each other. Things that are the same between these different genomes are probably going to be really important for how the virus works, and things that are different are going to be important for the differences that you have between the viruses from different places. And so this is an overview of some of the samples we've collected from Kamchatka. This is here, again, Kyushu in Japan, the first virus we worked with. Number of viruses from Iceland. Actually, I didn't collect any of these. Couple of viruses from Yellowstone. I collected this one, and then a couple from Lassen Volcanic National Park, which is our main hunting ground as well. Compare all these guys to each other, and what you find is a bunch of these open reading frames are present in all 10 of the genomes we analyzed here. Now we're up to 15, and a bunch of them, particularly this region down here, which was all green. Remember, you could knock them out or do insertions in here. The virus is perfectly happy. It's also not very well conserved relative to these other viruses. So this also gives us an indication what's going to be important for function. And interestingly enough, at least one of those genes up here in this region, which is otherwise present in almost all of these viruses, is not present in that many of them. We can knock this gene out, and the virus is perfectly happy. So these two ways, looking at conservation across the globe and changing things in the lab, actually correspond quite well with each other. Yeah? There are, well, it depends on how you count them, but anywhere from 34 to 36. You'll notice there are a couple of funky overlapping ones here. So as an overview for this, okay, what do we see? About half of the genome is what we call conserved, i.e. it's shared within a lot of these different ones. Not all the genes are essential, which we showed through these knockout experiments. And we also have some other examples which say the closer you find, so these two viruses from Lassen are actually more similar to each other than they are to viruses from Kamchatka, which are then more similar to each other than those that you find in Iceland. So there's an interesting connection between these two. One of the questions that immediately comes up, what's the host range of these guys? This depends on understanding an assay, which lots of people in my lab are getting really good at doing. So basically, this is a modified plaque assay. Good review for way back again, beginning of the class. Nice closing the circle here. You take a culture of uninfected cells, you mix it together with a soft layer containing media. We can't use agar because agar has problems at 80 degrees Celsius. So we use something called Gelrite. You put these two together, pour them on a plate, and then spot on the plate various different viruses, maybe from different parts of the world, different kinds of mutants which you've made, what we've looked at before. It's a lot of how these red arrows and green arrows come from. And then you just let the plate solidify, grow. Everything in brown here, all of our Sulfolobus. If you have something that would be like a red arrow, doesn't make virus, it doesn't cause inhibition of growth. If, however, it does, then we have what we call a halo or a clearing on the plate. So this is a great way to look at different mutants, but also a really good way to look at host ranges. And so this is the work of Caleb Marceau, ex-undergraduate student, graduated in 2009. Now he's finishing up his PhD at Stanford, where he looked at just these spotting assays of various different viruses across the top here. SSV1 again from Japan, 2 and 3 from Iceland, Lassen from the US, K1 for Kamchatka, and then he just compared them against various different Sulfolobus isolates from different parts of the world. These 3 are from Italy, these 2 are from Iceland, these 4 are from Kamchatka, these 3 from Japan, and this one down here from the US. You'll notice we're great hosts here. Now, USA, we're great hosts for all these different viruses. The Italians are good hosts, but the Russians are nowhere near as good hosts, and the Icelandics are not so good hosts either. What we had actually predicted was that there would be a nice geographical distribution whereby, say, a virus from Japan would maybe infect cells from Japan. Actually, it doesn't infect them at all. Or a virus from Kamchatka would maybe infect all these Kamchatkan isolates, but not some of the ones from different parts of the world. Turns out this is all over the map. So again, this virus from Japan infects some cells from Italy, but not those from Japan. This guy from Kamchatka infects practically everybody with the exception of a couple from Japan and Iceland. So it's really all over the map. So we have a lot more work to do, which is great. Keep people in my lab occupied for a little bit longer. So this is our review of what's happening with these Fuselloviruses. They have an integrase gene, which, as I didn't mention, but those of you who are watching carefully will have noticed that was one of the genes that was completely conserved across all of the different viruses. But we can knock it out, and the virus is still happy. So what's going on there? Turns out that Adam also did a nice experiment where he compared the 2 viruses, virus plus and virus minus, and competed them with each other in a lab, and he found that the virus that had the integrase gene won very quickly, which is exactly what you'd expect looking at the environmental data, or at least environmental genomes, where that gene has been well conserved. So evolutionarily, it is important, but in the lab, it isn't. And you also have to be very careful about this and the experiments you look at in any virus in terms of what you see in the lab. How much does it really relate to what's going on out in the environment? Lots of genes that don't match anything else in the databases, many of which are not required. You find these guys all over the place. Host range is kind of a mess. The SSVK virus that I mentioned on that last slide seems to infect practically everybody. So it's sort of a super virus. Turns out it's a real pain in the neck to work with, right? Probably because it infects so many of these different hosts, and actually is much more virulent than a lot of the other viruses as well. So now I'm going to switch gears and do very much a quick overview of some of the other viruses that are present in these environments. I mentioned these long filamentous viruses before. Again, they've got these nano-sized claws at the end, which seem to be important for receptor interactions. They've got double-stranded DNA genomes, but this double strand should probably be in quotes here, because again, it's something a lot like what you see with the pox virus genome, some of these giant virus genomes as well. Just a covalently closed end to each of the genomes. So double-stranded, linear, with covalently closed ends. Also have inverted terminal repeats at the ends of their genomes, a lot like what we see with the pox viruses and with the adenoviruses. What's a little bit different is that there are these repeat structures at the end that almost look like telomeres. And so whether there's some kind of telomere-like activity, so these are 5, depending on which one of the isolates you look at, 5 to 6 nucleotide repeats. This come one after the other, after the other, after the other, after the other, which again may be important for protecting the ends of these genomes. There are a number of different isolates here. SIFV, so FV just stands for filamentous virus, so flexuous rod here. SI is Sulfolobus islandicus, and then there's an Acidianus, which is a close relative to this. Again, cartoon-wise, this double-stranded, linear, or covalently closed end genome with these hooks at the end of the genome. So these are fascinating structures. Even more fascinating are these bottle-shaped virus structures. These guys were isolated again from an Acidianus culture. Acidianus is extremely similar to Sulfolobus. In fact, when you try and isolate Sulfolobus, sometimes you end up with Acidianus instead. These genomes have protein-primed replication, so this should be similar to what viruses we've talked about so far? Protein-primed replication. Polio is one, yes. What else? DNA viruses, protein-primed. Also with inverted terminal repeats. Not polio. I already heard polio before. Does that have inverted terminal repeats, polio? It's a single-stranded RNA virus. No. So inverted terminal repeats is going to have the same sequence on opposite strands of your DNA, right? So which adenoviruses have protein-primed replication? So not surprisingly, like adenovirus, these also have DNA polymerase genes. People were really excited about DNA polymerase genes in the genomes of these viruses. Why would they be excited about DNA polymerase genes in these kinds of viruses? PCR, because they're stable at high temperatures. So people looked really hard to try and find DNA polymerase genes in here. And also, these DNA polymerase genes from the viruses have a tendency to be relatively small and very stable, even above and beyond them being present in one of these extremophile environments. Turns out this protein priming is a bit of a pain if you're trying to do PCR, but that's another issue. Again, they've got these amazing filaments present here at the top. You can see the arrow here. There are 20 of these arranged at one end of the virion. What people immediately thought when they saw this is, oh, these are going to be interacting with receptors, but probably it's actually the other end, which is receptor binding, where these guys are associated with each other. Yeah, these are just amazing. These virions are absolutely incredible. Turns out that these are unfortunately really, hard to work with. So they haven't followed up more than just a couple of these really amazing electron micrographs here. But should I tell you a secret? You see things like this in Yellowstone too, and actually a whole bunch more of them. So I'm pretty excited about maybe following up on some of these things. We shall see. I'll keep you posted. So come back next year to virology, and then we can talk more about it. We also have these, what people call globuloviruses. These are blobs, basically. These should look extremely similar to a lot of the enveloped viruses that we've looked at so far. Again, don't have a really obvious structure, have envelopes around the outside. But the most interesting things about these viruses are that they are still linear, double-stranded DNA with inverted terminal repeats, but strangely enough, almost all of the open reading frames are all in one strand. It's really bizarre. If you look at where all the open reading frames are, they're all pretty much on one strand, so they're probably all being regulated by one set of promoters down at one end of the genome. Not very much work has been done on these, but it's a really fascinating kind of genome structure that they have. And again, that will be a whole lecture in and of itself when I teach my Archaeal virology class. Acidianus two-tailed virus. These are what they call the B-cauda viruses. Oh, by the way, I'm referencing all the things down here at the bottom, so if you want to go back and look at the original papers, they're all here. Probably the most fascinating thing about the Acidianus two-tailed virus and why it got published in Nature in the first place is that these guys, when they're being produced, actually look a lot like the SSVs, the Fuselloviruses that we talked about before. They're actually considerably bigger than the Fuselloviruses. Here's a cell that they're infecting. Here are those individual virions present on the outside here. These are the virions that are produced, but these virions are not infectious. The only way they become infectious is if they start to look like this. They have these two tails at either end. Well, how do you go from something like this to something like that? Well, it turns out that these virions undergo a morphological change outside the cell whereby they basically grow these tails. And this is what's shown down here. If you put these at 70 degrees Celsius, they grow these tails and become infectious. If you leave them at low temperatures, they just sit like this. So it's an extracellular maturation process. Very, very rare among viruses. And one of the things is a little hard to see here, but it turns out that the volume here stays absolutely identical. It's just a conformational change that happens. You have proteins present here in these spindle-shaped particles that then polymerize to make these long tails and basically push these hook-like structures at the end, which appear to be critical for infection, out to where these guys can now be infectious. Again, here they're not infectious. They have to have these extracellular maturation in order to become infectious and get into the cell. We've seen a lot of particles like this under the electron microscope. We had no idea what they were doing. And then this other group, led by a good friend of mine, ended up figuring out exactly what was going on here. Incredibly cool structure. Again, not clear quite what's going on there. It's got a large genome and a number of repeated sequences in it. So that's the two-tailed virus. I think this is the last main one that I wanted to talk about here, are the Rudiviruses. This is probably the second best studied of these Archaeal viruses. And this is mostly work that's been done by David Prangishvili's lab, and I'll show you a picture of David towards the end here. These are the Sulfolobus islandicus rod-shaped viruses, and here are some of these individual virions here. They look a lot like tobacco mosaic virus in terms of their structure. Just look at them under the electron microscope, with a couple of differences. The major one is that they've got this structure of 3 tail fibers at each end. Here's one end, and there's the other end. Here's the cartoon down here at the bottom. These are not enveloped viruses. It's just protein that's wrapped around the double-stranded, linear, with covalently closed ends genome. And one of the things that you see with these unenveloped helical structures, the longer the genome, the longer the virion. So it's not surprising, a lot like what we've seen in some of the negative strand RNA viruses. A certain number of nucleotides are bound by each of your individual subunits. So that is how you get the length of this particular particle. These have closed hairpin ends, again, a lot like some of the giant viruses that we talked about before. One of the things which is really bizarre, however, about the ends of this genome is that they seem to be base paired all the way along. So you've got TT on one strand and AA on the other strand. There's not enough space in normal DNA to be able to switch from one strand to the other between one phosphodiester bond, because they're about 2 nanometers apart. One phosphodiester bond is less than a nanometer. So how do you get from one strand to the other? Very interesting question about the ends here. Really don't know what's going on with them. One interesting aspect about these particular viruses, one of the reasons we know that the structure is exactly as long as the genome is, they have very high mutation rates, actually probably among the highest mutation rates for double-stranded DNA viruses that anyone's ever found. Quite why that is not entirely clear. Again, what, not why questions to ask in biology. And there are quite a few of these. It's not just Sulfolobus, but also Acidianus viruses as well. So one of the reasons that I wanted to talk more about this is it turns out that, let's see, today is June 1st. This was published May 22nd, is a cryo-EM structure of these particles, looking at how those proteins are actually associated with this double-stranded DNA. So here's a cryo-EM reconstruction at about 4 angstroms. A little hard to see, but here are your individual SIRV virions here. You can take these and literally chop them up into segments in the computer and then average each of those segments on top of each other. And since those segments are all identical to each other, you converge on a particular structure. And so that's basically what's shown here. We've got 2 monomers of the major capsid protein associating with the DNA here. This is a blow-up of what those guys look like. The big surprise when people looked at this, and again, literally this was Science a little over a week ago, is that the DNA, which is packaged here, is an A-form DNA, not B-form DNA. And the only way they know this is when you look at the electron density and you try and put B-form DNA into it, it doesn't fit. But if you just tweak it a little bit and make A-form DNA, which might help a lot with closing the ends of your genome, because in A-form DNA, your base pairs are really skewed relative to each other. They don't have the end structure here because they're having to average a whole bunch, so you don't have the ends. But here, at least in the middle, we've got A-form DNA, and this should hopefully not look like a normal double helix here. And then each of these different colored, mostly alpha helical proteins here represent the individual coat protein structures. So those bind to the A-form DNA, squish it together, and then also stack on top of each other in these structures, which again, when they all stack on top of each other, end up giving you this structure. Now remember, these virions are stable at 80 degrees Celsius and pH 3. So they've got to be very good at protecting this DNA from getting degraded and from, again, allowing it, once it gets in, to be able to be released and replicate, etc. So there's going to be 2 ways to protect your DNA: positive supercoiling, which is what we see with SSVs, and this A-form DNA, which is found in the SIRV structure, and literally just found a couple of weeks ago what that looked like. So we've got, oops, this one disappeared here. This is actually something that looks like a single-tailed virus here as well. In fact, very close to that picture I showed you right at the beginning of me sampling from that hot spring in Yellowstone, we found a number of virus-like particles. These are, I think, probably 2-tailed viruses that we didn't figure out at the time. This was in '99. In 2000, I was looking down the microscope and I saw these guys and I said, oh my God, these look like icosahedral particles. No one's ever seen icosahedral particles of these Archaeal viruses before. And then you've seen this picture before. Together with Jack Johnson's group at the Scripps Research Institute, we did cryo-electron microscopy, pseudo T equals 31 icosahedral structure, totally cool projections here at the 5-fold axis. And then Jack and Liang said, hey, that's not the interesting stuff. The interesting stuff is actually what's between these guys. And what's between these guys are the hexameric structures, which it turns out are really similar in bacteria, in our virus, but also in viruses like adenovirus, etc. So similar, in fact, these capsid protein structures, the major capsid protein, has no sequence similarity whatsoever between these 3 completely different groups of organisms, but their structures are practically identical to each other. So here's the structure for a bacterial virus. Here's the structure for adenovirus, and it turns out PBCV1, that algal virus that we talked about last time, also has a practically identical structure. This is the cryo-EM structure here in blue of our virus with this is the archaeal virus now with the bacterial protein. You notice it fits in here perfectly. How could this have happened? Could have been horizontal gene transfer, but that's horizontal gene transfer from bacteria to eukaryotes to archaea. Seems highly unlikely. Is it convergent evolution? So this is the best way to package things? Well, hopefully I've been beating over the head that icosahedra are a pretty darn good way to package a pretty large amount of nucleic acid inside a relatively small number of proteins. So it's certainly a possibility. We do know that you can make icosahedra with different structures here, but evolution, could be the way it's going on here. We think it's evidence for a common ancestor of all these viruses, but a common ancestor of all these viruses would presumably have been infecting the organism which is the last universal cellular ancestor to all of us, probably billions of years ago, which we tried to sell to Science and Nature and they didn't like, so we ended up publishing it in PNAS. So these are the structures which have been published a little bit later on. This is now the structure of the major capsid protein from our virus of Archaea, overlaid with these viruses, so say major capsid proteins of viruses from bacteria, and two different eukaryoviruses here in yellow and green. And this is what that particle looks like. Also has an internal lipid layer, which is interesting because that's also what's been seen in some of these bacterial viruses. But also, if you remember PBCV1 from last time, PBCV1 has a protein shell on the outside, nice icosahedral capsid, with a lipid membrane on the inside. So this internal lipid seems to be something which is also spread throughout a whole bunch of different viruses from a bunch of different domains. And in fact, those of you who remember when I went to Finland, again, way back when, the PhD defense that I was on was talking about a new bacterial virus that also had a structure very similar to this with a lipid membrane on the inside. And again, a bacterial example of this. So these guys look like they're really old, but there was another surprise in terms of these viruses, and that had to do with not this cool structure, not this really fascinating structure that's probably important for infection, but at the very end of the virus replication cycle, and that's how these virions get out of the cell. So we've talked about a little bit about budding, sort of budding at the membranes. We talked about lysis, where you break open the cell completely. These guys have apparently come up with a completely different mechanism, which of course is now disappearing here. These are what are called the virus-associated pyramidal structures, which hopefully we'll be able to see here. Yes, here it is. So both, actually back up here. So I see the pictures in here and then they disappear when I move to the next slide. Very strange. STIV, so Sulfolobus turreted icosahedral virus, again, not surprisingly, boring name for this amazing virus. But it turns out that SIRV, those rod-shaped viruses as well, actually have exactly the same kind of structure, which comes together to make basically holes in the membranes of these hosts. And what it does, it forms this seven-sided pyramid structure up here. 7 sides is very bizarre in terms of biology. But that seven-sided structure forms. You have a virion that comes underneath it, and then this opens up like a flower, and the virus particles are released. Really absolutely fascinating structure here. This was published this last year in the Proceedings of the National Academy of Sciences, where you can see the next really nice picture, which also has disappeared on here. This is a tomogram of a virus-infected cell, and all of these virions actually coming out of these structures here. Again, those of you who printed out the notes, hopefully will see this. It's a really amazing structure which is there. Put all this stuff together. What does it tell us about virus evolution? Well, we think, these are now our bacterial viruses, here are eukaryotic viruses, here are the archaeal viruses, sort of in between. I mentioned that a lot of the Euryarchaeal viruses look a lot like bacterial viruses. There may have been some transfer between these two, or there may have been some kind of common ancestor back here, which has led to the development of the viruses infecting these guys. In Archaea, we've clearly got a whole bunch of very different looking virus particles. And then the eukaryotes, for no apparent reason, seem to have a whole different class of viruses that are associated with them as well, certainly at the virion level, and how all of these guys are related to each other. So we think that studying these viruses may allow us to learn something about how these viruses function, but also just something about how viruses developed originally way, way back. So our summary slide here, Euryarchaea, mostly look-back bacteriophage, at least the thermophilic Archaea. Recently, people have found that these Archaea are actually present all over the place. They're present in soils. They're present in the oceans. They're actually maybe even some of them present in our guts. But no one's been able to isolate them. No one's been able to find viruses that associate with them. Most of those studied, certainly in my lab, but there aren't many labs that are working on this, look at the Fuselloviruses. These are incredibly widespread. Pretty much any hot spring in the world that you find has the appropriate conditions, temperature above 70 degrees Celsius, pH of 4 and below, is going to have these viruses associated with them. The genome is partially conserved, and we have a really good handle on this genetically in the lab, and we even think it's well enough prepared to work on in teaching labs. And those who are interested in the teaching labs, there'll be presentations on Wednesday afternoon. Come check them out. Down at the CLSB, what's it, 1N202, I think? I forget what the lab number is. But there'll be 8 different lab groups talking about some of the mutation work that they did on SSV1 here. There are a bunch of new different virus families that have been discovered in these Archaeal viruses. The Rudiviruses, which are those rod-shaped viruses that are encapsulating A-form DNA. The Lipotrix viruses, which are the long, flexible ones with the nano claws at the ends, probably have similar structures to these Rudiviruses. The Ampullaviruses, which are these guys, again with the birthday candles at one end. It was my daughter's birthday earlier this week, so think about that. But probably interacting down at this end with whatever receptor it is. Stay tuned on these guys. Hopefully we'll be working on them relatively soon. The B. cauda viruses, those are the ones that grow the tails outside of the cell. And then these Turiviruses here, which I was the first one to discover, which have the beautiful infection processes here, but probably more fascinating has to do with the evolution. Probably these are viruses related to the last common ancestor of our icosahedral viruses. And then they have this really amazing maturation and exit process through these 7-fold pyramid structures which escape on the outside. I can't claim all the credit for this. In fact, a lot of the work that's been done on everything except for SSV has been the work of David Prangishvili, really amazing scientist who used to sit right next to me during my postdoc in Munich, and has now moved on to the Pasteur Institute in Paris. Mostly he and his lab have discovered, say, the vast majority of these other new viruses. The work in my lab has been supported by the National Science Foundation, NASA, our friends here. I started all this work when I was working in Wolfram Zillig's lab in Germany, again together with David Prangishvili. Continued some of my work with Mark Young at Montana State University. Montana State University is a great place to be if you want to study thermophiles because it's right next to Yellowstone, only about 2 hours drive away. The Thermal Biology Institute there. Worked together with a couple of graduate students, Blake Wiedenheft and George Rice. Blake Wiedenheft is now working on CRISPRs. I don't know if any of you heard about CRISPRs. We haven't talked about that so much in this class. Fascinating subject that, again, something we could go on for ages about. And these are some of my other collaborators that I have there. If there are any more questions, you can ask them now. This is what Lassen Volcanic National Park looked like a few years ago. This hot spring has actually gotten considerably bigger since then. This has fallen down because the Earth has collapsed right near there. My website's totally out of date, but at least it's got some more information on it that you can check out there. So next time, Luis rate us. I'll be talking about virus ecology. And then on Friday as a review, next Tuesday, 8 a.m.,